Hi everybody, I'm Margaret Smith. I'm the corn breeder here in plant breeding and genetics at Cornell University. We have a very applied field corn breeding program and a tiny bit of sweet corn breeding that we do. So I'd just like to tell you about our program in general and then a few specific illustrations, since our time is short, of some of the things we're working on right now. New York State is a state where dairy is the primary agricultural enterprise, and one of the primary feedstuffs for that dairy is corn, both grain and silage. So field corn is one of the most important crops in the state. It occupies about 1 million acres of land here in New York. And we try to breed field corn in my program for what I would call niches. So New York State is a bit of a niche compared to where most of the corn production in the U.S. is, because we have a shorter growing season. We tend to have weather like this, not in July, but this year it's in July. Wearing a coat in the field in July is a really unusual event. But in the fall particularly, our weather tends to be very cool and wet. So we have some unique challenges during the harvest season that you don't see in the main parts of the central corn belt. And in the spring, our ground tends to be very wet. So we need varieties which will work with cool, wet soil conditions in the spring and lots of cloudy, wet weather in the fall. So that's some of what makes New York State corn a niche. Another niche we work on is organics. We have a lot of organic growers, and the organic acreage is increasing. So we do a bit of breeding work for organics. And then the other thing that I do to complement both of those is a lot of work on developing sources of resistance to diseases and insects. So that's the kind of overview of some of our major breeding objectives. We have a breeding nursery site here in Aurora, New York, which is about 35 miles north of Ithaca. It's a major corn growing part of the state. If I were to grow my breeding nursery near campus, not only is it a little even shorter season than what we have to deal with here, so there are a lot of things I couldn't effectively cross or work with because the growing season is too short there, but also it's not a major corn growing area, so I get every deer, raccoon, student needing a decoration for their porch, and everything else taking a big toll on my fields. So we grow our corn up here in Cayuga County, which is one of the larger dairy and field crops producing counties in New York State, surrounded by lots of acres of field corn to dilute out all of those deer and raccoon and bird problems and so on that we otherwise would have to deal with. So our breeding nursery is here at Aurora. In addition to the breeding program, we have a variety testing program that involves sites, 7 conventional on-farm sites, 2 organic sites, and those are all focused on grain testing, corn testing for grain. We also do both commercial corn testing for grain as well as for silage. So we have 4 additional silage testing sites that we run here in New York State, together with a team of folks from animal science and in collaboration with colleagues in Vermont. So we have a Vermont, New York corn silage testing program, which is nice because it picks up on a lot of the similarities of those growing environments, and the fact that Vermont also is a very big dairy state. So it gives us a broader sampling of sites, a nice collaboration that we can do for the commercial variety testing between those 2 locations. So that's the quick overview of the scope of our program. I want to start by talking about a little of the work we've been doing on organic corn breeding. And it's always unclear what makes a variety perform better in organic conditions than another. It seems fairly apparent that not necessarily the best varieties in conventional growing systems will always be the best for organic growing systems. And you can imagine a lot of the conditions that would make that true. The organic growers tend to plant later because they're having a legume or some other kind of cover crop plow down before they can get in the fields. They plant without commercial synthetic seed treatment chemicals, so germination and establishment is really important. They do a lot of aggressive weed control, so you have to have a variety that will withstand that. But translating that kind of thing, those speculations as to traits, nitrogen would be another one. Nitrogen availability is a more gradual process compared to synthetic nitrogen, which you pour on some as a starter and a bunch as a side dress. It comes in big flushes. For the organic corn, nitrogen is a more gradual, slow to start in the spring, stretched out over the season availability. So you can imagine a lot of things that would cause a variety to be better adapted to that than to conventional and vice versa. But most of those are speculative, and they're hard to translate into specific traits that I'm going to look for as I go through my nursery and say I'm selecting for this because that will make the better organic corn. So we do a lot of our selection for organics based on variety evaluation in organic systems, looking at what works well, trying to make new combinations among some of the best performing things and reselecting those. Sort of standard approach to trying to improve adaptation and yield performance in a corn breeding program. Figure out what works, try and combine things that work well to get something even better. One of the things we've done that's a little unique in our organic breeding effort is I worked with colleagues, Kevin Montgomery out in Illinois and a colleague of his, to do cold stress testing on seeds and seedlings. So I mentioned that we have cold, wet soils in the spring, that the fields need to be able to establish and get going without the benefit of a seed treatment, and that they need to establish and get going quickly because you need canopy cover to help with that weed control problem. Managing weeds is one of the biggest challenges in an organic system. So we took segregating populations, so these would be F2 populations, subjected them to a cold stress test. So put the seeds in water to hydrate them, then put them in cold temperature conditions for several days, then moved them to germination, petri dishes or paper towels for germination, took the ones which germinated well and planted them to the field, and that whole part was done by my colleague out in Illinois and his collaborator, planted them out in the field and self the things which happened to grow well despite having been mistreated in that way as seeds and seedlings, and then we're doing breeding with those. So one of the things that the progenies I'm standing in front of happen to be things that have emerged as a result of that. We screened them as S2s, they were selfed as S3s, we test crossed them as an early generation test and put them in an organic yield trial and took the best of them. These are now S4s, we're advancing and test crossing again to see whether that gives us a fraction of that original population that has better establishment, better tolerance to cold, wet soils, more rapid early vigor and canopy closure to help with weed control. So I'll be trying to answer the question of whether that subset of the F2 is a better performing subset in organic conditions than if we had just used our standard approach to early generation breeding and selection. Our entire nursery here is done without seed treatment, but the challenges in a conventional nursery like this are less than what you were going to find in an organic nursery or an organic grower's field. So we did start these off in the early generations in our organic breeding nursery to keep that selection pressure on, and we just like to compare whether they're going to really help us, because the cold stress test effort, as you might imagine, stressing seeds by pedigree and putting them in germination plates and hand transplanting them to the field is a fairly labor-intensive process. So we'll see whether that's likely to work or not. Another area I'd like to talk about is our disease and insect resistance breeding. So this program historically has done a lot of breeding for resistance to various corn pests. And it fits well into our adaptation, both for conventional and organic. So it feeds into those programs, and all the progenies that come out of it go right into our testing program. So I want to take a few moments to talk about several of the different pest resistance efforts that we have ongoing. One of the diseases we've worked a lot with is the anthracnose stalk rot, and the progenies here on my left are some of the results of some of that selection. So anthracnose stalk rot is a atypical stalk rotting organism. Most of the stalk rots in corn attack the stalk when it's senescing, so the plant is already on the way out, the tissue is dying, the rot moves into that dying tissue, so we call them stalk rots of senescence. Anthracnose is not like that. It actually attacks stalks when they're still healthy and vibrant, so at around flowering time or a little before. And what that means is that stalks are also critically important still in terms of translocation of water and photosynthase during that time period. So the fact that the rot is attacking and moving through the stalk at that early time in the plant's development means that it has much more potential to cause yield losses. It's cutting off photosynthetic translocation to the ear, it's restricting movement of water and nutrients up from the soil, so it can be really problematic, in addition to weakening the stalk. And you may remember at the beginning I said we have all that cloudy, wet weather in the fall, which means our plants have to stay standing in the field oftentimes longer than farmers would wish. Sometimes they're just not maturing very fast because of the cool, cloudy, wet weather. Sometimes they're mature enough, but the farmer can't get near them to harvest because the ground is too wet. So stalk quality and standability, despite that, are really important to our growers out here in the Northeast. So we've worked on selecting for resistance to anthracnose stalk rot. I have a notable memory from speaking about this to a seed industry meeting and having one of the seed industry guys raise his hand and say, Margaret, you know, that all looks really exciting, great resistance levels, but make sure you're not selecting for resistance to yield too. Because this is what often happens. We can develop really resistant things, but they're really resistant maybe because they aren't even growing an ear. There's nothing drawing the nutrients out of the stalk to begin with, or just because they seem to take a big yield hit. So our anthracnose stalk rot breeding program we have run by doing every generation along the way. We'll take our selected progenies that appear to have stalk rot resistance, cross them to a stalk susceptible tester to evaluate that resistance in a hybrid combination to make sure they're transferring resistance to their hybrid progeny, and simultaneously cross that line to a good yield tester so that we can look at those hybrids and make sure we're not breeding for resistance to yield. So the progenies on my left here are some of the ones that have come out of an effort that started in the Germplasm Enhancement of Maize program, which was a federally funded effort to enhance the diversity in commercial maize germplasm. It was a collaborative effort between the corn seed industry and the public sector breeders and a variety of hubs where the GEM, or Germplasm Enhancement of Maize program, was centered. So we got some funding early on to work with that. We identified progenies. The one that I'm standing next to happens to have come from a variety that came from Chile, but varieties that had come from the germplasm collections, largely from the tropics and subtropics, that are hugely diverse in maize, but have not been widely used in commercial breeding. So these are some of the things emerging from that breeding effort for anthracnose stalk rot. The resistance is looking really good. We will inject these right into the base of the stalk with stalk rot and split the stalks at harvest to evaluate how far the stalk rot has traveled up the stalks, make sure we're selecting for resistance. And then in the background behind me, the stuff that's really tall, you can see, are some of the test cross hybrids that are being evaluated for anthracnose stalk rot resistance. So as I mentioned, each generation of the way, S2s, S3s, S4s, we are crossing simultaneously to both a rot susceptible tester to evaluate the contribution of this progeny to the stalk rot of the hybrid, and that's the hybrid evaluation going on behind me, and at the same time to a yield potential tester to evaluate its contribution and adequacy and adaptation for yield and productivity here in New York. So that's some of our anthracnose stalk rot work. One of the other things that acts synergistically with the anthracnose stalk rot is the European corn borer. There is less of a problem with that now because of the widespread use of Bt corns, but there are still a number of circumstances where growers may not want to use or be able to use the Bt as a control mechanism for European corn borer. So you can imagine organic growers are not using those things, and growers who are planting Bt also need refuge acres that are non-Bt. Thirdly, I maintain that having some innate corn-based resistance in the background of Bt will probably help to ensure the longevity and the effectiveness over time, the durability of that Bt-based resistance. So for all those reasons, we've continued to work on European corn borer resistance, and the European corn borer has been shown to act synergistically with the anthracnose stalk rot. So if you have corn borers creating holes in the stalk, you have entry points for the rot. There is some evidence that the corn borers can even vector in the anthracnose. Anthracnose will cause a leaf blight disease phase, which can produce spores that can then be carried in by the insects. Incidentally, resistance to the leaf blight phase of the anthracnose disease is genetically unrelated to resistance to the stalk rot phase, so that makes selection particularly challenging. But the corn borer can act synergistically. So we've had a long-standing relationship with several international programs that select for borer resistance, most notably the one at the International Maize and Wheat Improvement Center, CIMMYT, in Mexico. CIMMYT developed a multiple borer resistant population that was selected all across the world, including here in New York, for resistance to a large array of different types of borers. Most of the tropical and subtropical borers are much more aggressive than the European corn borer that we primarily deal with here. So having things coming out of that program provided really strong levels of resistance to stalk boring by the European corn borer. This is another pest where the leaf damage phase and resistance to it is genetically unrelated to resistance to the stalk boring phase. So these are the two classic problems. You can select for leaf beating resistance, but it does nothing necessarily for stalk boring resistance. And as you might imagine, stalk boring damage by this insect has the potential to cause much more yield loss. You've got tunnels through the stalk, all the same reasons I talked about for the anthracnose stalk rot. Tunnels that cut off translocation through the stalk is weakened and it can fall over, so all of the same reasons. So we took some of these materials from the multiple borer resistant population in Mexico, and also the one that my former colleague, who I miss very much, John Mim, the entomologist at CIMMYT for many years, deemed the BRG. If you worked with John at the time, you would know that BRG stands for Gordo Resistente Gringo. So he called it that because he had planted some U.S. germplasm, that's the gringo part, adjacent to the multiple borer resistant population, and he harvested a bunch of top crosses off of that. So it was multiple borer resistant by U.S. corn belt germplasm. So that's the BRG. So this line right to my right is one of our progenies that is emerging from that breeding effort with the BRG. We're again selecting for insect resistance every step of the way as we inbreed these, making test crosses both to susceptible testers to evaluate whether the inbred conveys that resistance to its hybrid progeny, and to commercial testers to evaluate its yield potential and adaptation and general productivity. Interestingly enough, we were struggling with crosses to these materials derived from the BRG. We kept having poor seed set, and I've been stewing for a couple years about what was going wrong with that, and finally the light went off, and I think they have a gametophyte factor. A lot of tropical genetic materials in corn have floating around in them gametophyte factors, which limit what can be crossed to it, what will pollinate it successfully. And I'm suspecting these have a gametophyte factor floating around in them. So we're making some controlled test crosses here in the nursery this year to evaluate whether that's actually the case. I'm thinking that's probably a GA1S. This is the same factor that's used in the white corn industry and the popcorn industry to prevent lots of cross-pollination to commercial crops in those arenas with standard US number 2 yellow dent. If you're trying to grow white corn, you don't want lots of yellow kernels in there. If you're trying to grow popcorn, you have this gametophyte factor to reduce the chances of pollination from adjacent field corn that would decrease the popping quality of your crop. So we're evaluating those for whether they do in fact carry this gametophyte factor, and we also have hybrid evaluations both for yield as well as for corn borer resistance going on with some of these new sources from the famous BRG. So the last thing I want to talk about today, I just finished talking about multiple insect resistance, and I'd like to talk for a moment about multiple disease resistance. One of the things that's emerged from our breeding program is a pair of inbreds, sister lines, that we had originally selected for resistance to gray leaf spot, which is one of the more problematic leaf blights in corn. And upon further evaluation, it turned out that they in fact had a fairly broad spectrum of disease resistance to not only gray leaf spot, but also northern leaf blight, some level of resistance to southern leaf blight, anthracnose leaf blight, rust, and even Stewart's wilt, which is a bacterial disease. They had some moderate resistance. So we got interested in the cause of that multiple disease resistance. And one of my graduate students did a fairly big recombinant backcross population to try and understand the genetics underlying that multiple disease resistance. It involved a lot of different disease evaluations, and paper should be coming out. It was just accepted, so paper should be coming out shortly on that. To cut to the chase, he found a number of different loci, several of which are in areas that have been identified as kind of hot spots in the maize genome for disease resistance. One of the things that emerged was the area that contains the ligulus 1 gene, which has also just been published as having some relationship to disease resistance. Ligulus 1 has to do with the formation of the the ligule and the auricle and that leaf blade sheath interface, the whole development pathway there. But it also appears to be linked through perhaps ligulus 2 to some regulatory systems that may control the salicylic acid pathway. So that would be a logical reason for it to be perhaps related to disease resistance. So we're still trying to understand this rather serendipitous multiple disease resistance we found. But one of the things I'm doing with it is trying to backcross it into sweet corn. So you may have noticed that this corn looks a little different from what we were looking at. This is sweet corn. So we are trying to do some generally slow, gradual backcrossing to see if we can move some of that multiple disease resistance trait into sweet corn. A lot of sweet corns, one of the primary things that they need is a good disease resistance package. That's the primary thing after the real primary thing, which is good flavor and quality. So we're focused on how can we piggyback some sweet corn improvement in a small way on our large field corn breeding program. That's by far the biggest part of my effort. And so this is one of the examples. So the progenies I'm standing in front of right now are a bunch of progenies that have been, we're doing backcrossing with selfing generations in between, and then we screen those selfing generations for several different diseases. You can imagine this takes a lot of field space and a bit of time to get through several diseases, and especially when you have the year where the anthracnose doesn't do anything, and the year where we don't happen to have any rust, or the year where we have way too much of one disease and you can't even see the rest of them. But we are slowly trying to move some of these genes into a sweet corn background. Probably from our program, that won't be a commercial sweet corn. That will be a source material that the industry or other sweet corn breeders could use in their breeding efforts. So there's a quick lightning tour of some of the major breeding objectives we're working on in my program. I hope you've enjoyed it, and I hope you have a great time attending all of the other talks that are part of this meeting. Thanks very much.
This has been a production of Cornell University, on the web at cornell.edu.